Goedenavond allemaal. Wat bijzonder, want wij beginnen aan ons 9e webinar. En we hebben deze keer niet 1 gast, maar we hebben 2 gasten aan onze virtuele tafel. Even een kleine check. Ben ik goed hoorbaar, Els en Arnaud?
Yes.
Yes, thank you. Allereerst ontzettend welkom. We hebben heel veel inschrijvingen, meer dan 626. Er komen heel veel mensen loggen nu nog in, zien wij. Maar het is ons 9e webinar. 100 jaar geleden is de insuline uitgevonden, maar dat is een behandeling, maar nog geen genezing. We gaan vandaag het ook echt over genezing hebben. Welkom Arnaud, Dr. Arnaud Zaldumbide. U gaat zichzelf zo meteen voorstellen en dan schakelen we over op het Engels. Els Anken, bestuurslid vanaf eigenlijk het eerste uur bij de oprichting van JDRF in Nederland. Ongelooflijk betrokken bij JDRF en alle ontwikkelingen. Wij zijn alle 3 vanavond uw tafelheren en heer en dames. Er stroomt nog heel veel binnen, maar ik ga wel de presentatie starten. En ik begin met iets heel belangrijks, want hier gaat het om. Het gaat om geld werven om uiteindelijk het belangrijke onderzoek te kunnen financieren. En ik wil meteen beginnen met onze walk die 19 juni aanstaande plaatsvindt in Museum Boerhaven in Leiden. En juist nu, we bestaan 10 jaar. Vorig jaar hebben we €127.000 opgehaald. Je ziet hier mijn vriendinnetje die de cheque heeft overhandigd aan ons destijds. Maar dit jaar opnieuw. We hebben een doelbedrag van €150.000, een streefbedrag. We zitten op ruim 67.000 en ik wil u allemaal uitnodigen van ga op je handen lopen, zoals dit meisje. Ga door de modderbroeten, zoals dit team. Ga wandelen of ga gewoon doneren, want dit webinar is gratis, maar het onderzoek is echt ongelooflijk belangrijk. En juist nu, in de tijden dat heel veel laboratoria toch vertraging hebben opgelopen, hebben we uw donatie ontzettend hard nodig. Maar goed, dat is 19 juni. Nu gaan we eerst het hebben over vanavond. En ik zal u heel kort meenemen in een aantal strategische ontwikkelingen betreffende de onderzoeksrichtingen vanuit JDRF internationaal. Daarna zal Dr. Arnaud Zaldumbide de presentatie verzorgen. En Arnaud, ik ga Arnaud hier in beeld. Arnaud, most welcome. You're coming from France originally, and you work very closely with other scientists all over the world. And how important is it that this international collaboration is here in the Netherlands?
I think it's a good evening, everyone, first of all, and it's a pleasure for me to be here. So I think it's essential, to have this large network of people, a large network of scientists from different fields also to share our expertise. So I think it's essential to find our type 1 diabetes developed and it's the first step for a cure for type 1, I think.
Yeah, thank you so much. So important is it that it's international organized, everything. So our mission is het versnellen van levensveranderende doorbraken voor het voorkomen, genezen en behandelen van type 1 diabetes en haar complicaties. En daar zijn we elke dag mee bezig. En afgelopen jaar heeft JDRF International opnieuw gaan kijken naar de prioriteiten. Dat is eigenlijk samen te vatten in 5 hoofdonderwerpen. De wereldwijde universele screening, wat heel belangrijk is om in een vroeg stadium type 1 te diagnosticeren voor de behandeling en uiteindelijk ook hopelijk voor de genezing. De disease modifying therapies, dit richt zich vooral op het voorkomen, stoppen en het terugdraaien van het verlies van de functionele bètacellen. De onafhankelijkheid van insuline, het verminderen van de ziektelast. En dat geldt zowel voor de fysieke ziektelast als ook voor de psychische impact, waar steeds meer onderzoek naar wordt gedaan. En ook het trainen van de onderzoekers, de researchers all over the world. Dat zijn eigenlijk steeds de 5 belangrijke richtingen Waarbij moet worden opgemerkt dat er steeds meer onderzoek gaat naar naar de oplossing. En daar gaat tegenwoordig al zo'n 75% van al het onderzoek gaat echt naar cure. Dr. Arnaud Zaldumbide, just introduced you a little bit. You worked together with Bart Roep in the past. Bart Roep is also in the webinar we saw. It's very special. A special welcome also, Professor Dr. Bart Roep in this webinar. Very nice to have you here also. I would like to invite you to do the presentation now. I will start up the presentation online, and later on we will have the discussion together with each other. We have already more than 50 questions in our mailbox, but we will see what we can do. The presentation is in English because we translated everything, so we can understand. Everybody can understand the presentation. So I will start the presentation now.
So good evening, everyone. So it's a great pleasure for me to be online tonight and to be able to share with you some of our results from our research. So as Ingrid said, I'm working at the Leiden University Medical Center, so not in the main building, but in the research building. And as you can see here, I mean, although we are doing fundamental research, basic science, we are still connected to the clinician via this glass bridge here on the picture. So we are always in mind to try to treat patients for type 1 diabetes. So before I start with our result, I'd like to start with this page of the newspaper in order to celebrate the anniversary of the discovery of insulin. So this in 1921, so 100 years ago, we celebrate this year the 100th anniversary. So it's a great discovery. It has been a fantastic breakthrough for the scientific community. It has also been a lifesaver for the diabetic population. But so the work from Banting, MacLeod and Best are also open to a cure of type 1 diabetes. But as you can see from this paper in 1922, the Toronto doctors were on track for diabetes cure. 100 years later, the cure is not there. So we are getting closer, but not there yet. And maybe us as a scientist, we should take that as a warning to see the disease from a different angle. So, as you know here, the insulin in your body is produced by this organ, the pancreas here in red, that's located under your stomach. And within this organ, with the pancreas, you have some microorgans that are called the islet of Langerhans. This islet of Langerhans contains endocrine cells that are secreting hormone responsible of the regulation of your appetite and your blood glucose level. Within this microorgan, two main cell types are there. The ones that are painted in green here are the alpha cells who produce glucagon, and those glucagon is responsible of the release of the glucose to the liver. The other one, the counterpart, is the beta cells, so the insulin-producing cells that are here in red on this picture. In type 1 diabetes, here's the situation. So you can see that there is a complete or almost complete destruction of the insulin-producing cells in type 1 diabetes. This picture also shows you something striking. It's also that the other endocrine cells within the islet of Langerhans are not affected by the disease. And this, to give you a definition of type 1 diabetes, is characterized by the progressive but also the selective destruction of the insulin-producing cells by the immune system. So if the end stage is very clear, the cells are destroyed by the immune system, all the scenario and all the molecular mechanisms involved in destruction are relatively poorly unknown. So here, if we want to describe what we know from type 1 diabetes and how things occur. So here I depicted on this slide, you can see the islet of Langerhans with the beta cells in green, the alpha cell in blue. You see that these islets are very close to the vascularization in order to allow a very fast release of the hormone in the bloodstream. And we know that in type 1 diabetes there is a first inflammation, a first event that will trigger the destruction of the beta cell. There is still a big question mark on this primary trigger. Several hypotheses have been proposed: viral infection, change in the microbiome composition. Nevertheless, following this first inflammation, there is a response of these cells that will start to secrete some mediators that we call chemokine that will attract immune cells and cause the immune cell to infiltrate the pancreas. These cells are dendritic cells, macrophages, but also CD4 cells, so T cells that will secrete pro-inflammatory cytokines, and another type of T cells that are the CTL, the cytotoxic T cells, and these ones are involved in destruction of the beta cells. Altogether, you see that you have the islet are swimming in an environment of immune cell that you call insulitis that precede the development of type 1 diabetes. What we do in the lab, we try to understand what are these T cells, so the cells that are really responsible of the beta cell destruction, and we know that these cells are able to destroy the beta cell because they recognize at the cell surface of the beta cell a fragment of a protein. So we try to understand what are the T cells and what do they recognize at the cell surface. So in order to explain you a bit more in detail what happened in this cell, I need to bring you inside the cell. So you know that all your cells contain nuclei. In this nuclei, you have the genetic information which is contained in one molecule called the DNA. This DNA is then transcribed into another molecule called the RNA that contains the information required for the protein expression. The proteins are there. And this information is read by a complex called the ribosome. So basically, the RNA carries the information, the ribosome reads the information on the RNA and translates that into protein. If we try to make a scheme in a simplistic way of this translation process, here you have an RNA molecule with carrying an information, and this information is carried by letters. One letter is what we call a nucleotide. A group of 3 letters will make a word, and we call that an amino acid, and a chain of amino acid is a protein. So it's like reading a book. And the ribosome is doing the following. So it sees the beginning of the sentence in this RNA information, bind to the start, and then assemble this group of 3 letters in order to form a word. The first word is assembled in the sentence. The ribosome will move forward to the RNA, assemble the second word, and this till the end of the sentence where the ribosome detaches and where the protein is formed. And you can see here on this simple example that the sentence or the protein made from the information carried by the RNA is grammatically correct. Now if we imagine that the ribosome reads the sentence starting from a random point or alternative starting site. You can see that a similar process occurs. The ribosome will see the beginning of a sentence, or what it thinks is the beginning of the sentence, assemble the first word, assemble the second word, and then ribosome release, and then you have a sentence here also. And as you can see here, this sentence, even in French, is not meaningful. So this sentence is an aberrant protein, aberrant sentence made from the same mRNA molecule. And here what happened, so aberrant molecules are theoretically recognized by the immune system as wrong protein, and the immune system is normally destroying a cell that accumulate a wrong protein. So this concept is not new, of course. You know that your immune system is there to protect and to maintain the integrity of the body, and is able to distinguish what is self, so all the cells that belong to your body, and the cells that are correct, the proteins that are correct, from proteins, so identified proteins that are non-self or aberrant protein. So this, for example, is what you see when a virus infects a cell. The cell will express some protein, viral protein, and the immune cell will recognize this viral protein as something non-self, and the immune system will try to destroy virus-infected cells. We also see the same thing when a cell is turning into tumor. A tumor will start to accumulate some translation error, aberrant protein, and in that case the immune system will try to fight against the tumor, so the immune system will try to target the cell that is turning into a tumor. So this concept we have tried to developed this concept in several reviews that I wrote with my colleague Bart Roep, where we try to demonstrate that maybe in type 1 diabetes, the immune system is doing what it's supposed to do, just destroying a tissue that have accumulated mistake. And in that respect, type 1 diabetes should not be seen as a problem from the immune system, but as a problem from the beta cell. I'll come back to that at the end. So, what we've done in the lab in this project sponsored by JDRF, I'll just show you some examples. So it's like the same process of the translation process, slightly more complicated, where you see the mRNA, the ribosome, and here it's the whole cycle of translation of RNA information into protein in a slightly more complex way. What we have been able to do is to freeze the ribosomes at the translation initiation phase. So in order to get a snapshot of the ribosome at the moment where they start reading the mRNA information. We have been able to do that, in that case, to freeze this ribosome, to remove the RNA that was not protected by the ribosome, to separate the ribosome from the RNA, and then we have sequenced all these small fragments of information containing the start site, so containing the beginning of the sentence. We have aligned that to the whole genome here in order to identify where, in the whole genome, where translation of the protein occurs. So the results were quite surprising for us because we realized with this experiment and with this kind of assay that what we know from the beta cell proteome, so what kind of the protein that are produced by the beta cell, represent only 17% of the non-protein. Lots of things are unknown. So what we see is indeed what we know from the beta cell is indeed the visible tip of the iceberg. We also have found that many of these proteins are only produced in inflammatory stage compared to normal situation. Altogether, we also have identified that plenty of proteins are differentially expressed, and most of them are characterized by the response of the cell to inflammation, the response to stress, and protein degradation. If we go now a bit more specifically on what happened when we look at the insulin mRNA, so the insulin translation, we need to realize that this, again, the islet of Langerhans with the beta cell in red, and what we know is that a beta cell is indeed an insulin factory. 20% of the RNA that I just talked about is dedicated to insulin production. And upon glucose challenge, a beta cell will produce up to 1 million molecules per minute and per cell. So it means that the whole process I just explained you of ribosome reading the sentence, elongation, protein synthesis occur 1 million times per minute in each cells of the pancreas. And indeed, when you do so many things at the same time, you may imagine or you can think that you can accumulate mistakes. And indeed, it's what we have described. So you can see here is the information, the mRNA of insulin on the right. You can see that we have no doubt about where the start site of insulin is the one frame here, there. So we can encode the pre-pro insulin from this RNA. And what we have identified is this RNA carries also an information for another protein here, different, completely unknown, but has no homology with all the known proteins, so part of the unknown part that I just mentioned. And this protein is indeed immunogenic. It's what we've described a couple of years ago in this paper published in Nature Medicine, where indeed the insulin mRNA can encode an out-of-frame polypeptide. To conclude here, I'd like to talk about the take-home message from this research that we are doing. First of all, we have discovered that the beta cell proteome is way more diverse than we think it was. We also have discovered that the translation is not flawless, so we thought that it was a perfect system, RNA information, protein, but not. There are errors that are made during this process. And although these errors are part of any biological process, they may have some dramatic immunological consequences. As an illustration, the protein that's encoded by insulin that I just described, we have found that in the blood from type 1 diabetic patients, there are some T cells, the CTL that I explained you at the beginning, that are dedicated to recognize this particular peptide, and much more T cells in type 1 diabetes than in healthy individuals. Also importantly, when we have tried to isolate these T cells from type 1 diabetic population and try to see how they react against the beta cell, we have found that they were responsible of the destruction of this beta cell, and this destruction was increased in high glucose and inflammatory conditions, so situations that mimic type 1 diabetes. So you see, one error is responsible for the destruction of the beta cell. So why is it so important to understand what happened and what is seen at the surface of the beta cell? I'd like to finish on this scheme so that I started with, where you see the islet of Langerhans in the middle of the immune cells during insulitis. And if in this scheme we try to see what kind of strategy have been used to try to prevent type 1 diabetes or to delay type 1 diabetes, you can see the surprising thing is that we have used anti-T cell antibody, we have used immunosuppressive drugs, we have used anti-cytokine treatment. All these treatments are focusing only on one compartment, which are the immune cells. Nothing has been done to protect the beta cells. And indeed, it's what we have tried to also convey as a message in a review that we recently wrote for Nature Review on Endocrinology, that type 1 diabetes is maybe a disease of the beta cell and not so much of the immune system. So our goal at the moment is to try to intervene on this beta cell, try to relapse the stress on this beta cell, and it's a bit also what we have described in a paper published earlier this year, where in diabetes here we have shown that inhibitors of ER stress prevent the T cell mediated destruction of the beta cell. So this is one possibility to intervene on the beta cell. Altogether, all our research by the identification of the peptide are extremely useful for better diagnostic of the patient, and by having a better diagnostic, we may do the first step for personalized medicine and some antigen-specific therapy or reversed vaccination to treat type 1 diabetic patients. So all this work has been done at the CCB department at LUMC with our collaborator for the LUMC, also in close collaboration with Bart Roep at the City of Hope in LA. And thanks for the support of JDRF. Without them, all this research would not be possible. Thank you.
Thank you, Arnaud, for this presentation. What a very interesting story. I'm really interested, Els, what is the most helpful, in your opinion, when you hear this presentation?
I think the most interesting observation is that there's been a kind of split in before the trigger that causes type 1 diabetes to manifest itself, which is where the immunologists look. And now my understanding that after the trigger, this is where molecular biologists like Arnaud Zaldumbide look. And this discovery of there being, he mentioned the mystery of the disappearing beta cells. And remember, beta cells are what makes the insulin in your body. And then suddenly they mysteriously disappear and no longer do what they were supposed to do. When you discover this, this means that where we're looking for the cure would need to be in a different place. But yeah, so that's the major observation. It's kind of like everybody is working in one place, and then suddenly with the research of Arnaud combined with what Bart is doing, we're saying, yes, another forest, please. Mm-hmm. And so this should open a lot of opportunities. I see that Bart is asking to give some comment or some translation. I don't know if there is sound for the audience.
No, that's not possible. But we, when Bart will ask.
If you could help in the chat,
yes,
that would be great.
Yes.
Yeah. We do have a lot of questions. Ingrid, is this the time to actually start taking some of the questions? Because many of you have been sending in your questions also when you signed up for today. And what we've tried to do is take the sequence of Arnaud's story and then actually try and see where we can answer most of your questions, because I must say many of these are quite similar. So we have a couple of groups of questions. One is the inflammation that you mentioned that causes cells to be stressed. There are several questions coming in from the audience regarding this stress. The word stress, of course, some people were saying, well, if there is negative stress or too much stress, what kind of stress are we talking about? Are we talking about stress like in your head? Is there a way you could prevent that by making the beta cells more zen? So one is, could you explain the stress and what people could do to take down the stress? Is there any way they can do that? And let's take that first, if I may, Arnaud. You are mute.
Yes. Arnaud?
You're still mute
now. No. Sorry for the moment. Can you hear us, Arnaud? Yeah. Okay. And I'm not unmute you.
Maybe you can use your phone, and then we'll put our phone.
And now it's better?
Yeah. Now
yes,
it's better.
It is.
Thank you.
Good. Sorry for that. So yes, first of all, thanks a lot for all these questions. So when we talk about stress, I think it's a very vague word, stress. So we have to distinguish two types of stress. What we describe at least in the research we are doing, is the cellular stress. So basically, we should see that as a kind of imbalance between the production of a protein and the secretion of a protein. And all this protein that is produced but not secreted accumulate in the cell and is a source of stress. So it's the case, for example, for insulin, what we described. So accumulation of this insulin production in something called the endoplasmic reticulum is a source of stress and apparently a source of increased immunogenicity of the beta cells. So this is for the cellular stress, but of course, I think as a patient, you're also very interested in the role of the psychological stress, and this is also another aspect that we are not touching upon in our research. But for example, there are lots of parameters that can affect the beta cell. In particular, for example, you know that in stress condition, your body is also secreting hormones. One of very important hormone of stress is the cortisol, and cortisol, for example, has been shown to play a role on your hyperglycemic condition. So hormone release during psychological stress will affect your glycemia, and this glycemia is also the source of stress for the cell, because upon high glucose, the cell has to produce more insulin. So just to show you that everything is kind of connected. As soon as the metabolism changes, the demand in insulin is changing because of increased blood glucose, because of hormones, because of inflammation. You create a cellular stress that may be prone to for destruction for the beta cells. So the stress is on several aspects. The stress, the psychological stress, the cellular stress, in theory, is not good. So yeah.
So if I collect what you say, if I summarize what you say, cells with less stress will help keep your blood sugar, your blood glucose stable as looking as a patient.
Yeah, as a patient, it's what we think is that less stress cells is less prone to be destroyed. So one way would be there is drug already available that will be able to relapse the stress. And by relapsing the stress, we may have cells that will be less destroyed. Another way to intervene on the stress is, I think it was another part of your question, since the high blood glucose level is a source of stress for the beta cell, a very good control of your glycemia when you're a patient is also a way to slightly remove some pressure on the beta cells. So one way to remove the stress is also to have a very good blood glucose control.
So you're actually going to go in an upward positive spiral. If you control your blood sugar in a good way, then you actually create that there is less stress.
Yeah, yeah.
And then the reaction towards the stressed beta cells is also less.
Yeah.
Is that the loop you're saying?
Exactly.
And a question that comes in from the audience is, how long is it then going to take before you were mentioning there are certain drugs that can also already be applied for this. People are asking, when will they be able to access that?
So I think it's a very good question. I think it's also very important for us as a scientist to realize that we are not moving fast enough to a cure, but we need to realize that there are lots of clinical trials that are ongoing. I think more than 500 that are recruiting patients that are testing drugs, including drugs to protect beta cells, including drugs that are relapsing stress, but also we'll probably discuss that later on about antigen-specific immunotherapy. So there are already some things not on the market, but as a clinical trial, at least to test the safety and the efficiency of the treatment. So it's already in progressing. The problem, I think, with type 1 diabetes, it's the type of population you will treat with these drugs. So you can imagine that all the intervention that may extend or reduce the stress on the beta cells can only be applied if there is still some beta cell left in the patient. So it will mean that all the drugs that are potentially available on the market to do this kind of effect have to be used in very early patients, or even maybe on patients that are not patients yet, on people that are at risk. So it's the difficulty to treat type 1 diabetes. It's to have an earlier intervention point to have the best chance to cure the people.
And that immediately also makes clear why immunologists and who are before the trigger and you who are right after the trigger need to work so closely together, which is exactly what you were describing. One of the questions that came in was, can you actually create new beta cells? And if you do, or if you transplant them, how long can they work before they get perished again?
So again, excellent question. So you know that in many cases in type 1 diabetes, all the beta cells are destroyed, or most of the cells are destroyed. In that case, the only therapy available will be to bring a new source of beta cells. So at LUMC, there are some work ongoing with pancreas transplantation, islet transplantation. These are therapeutic approach that are ongoing at the moment, but the future is moving to stem cells and to beta cells made from stem cells to have a source or unlimited source of beta cells. Yet, I think you define very clearly the problem. If we bring new beta cells in a body where there is already an autoimmune reaction, the chance that these cells are destroyed are, of course, very high. So that's why it's very important to work, for us as a molecular biologist, that we can make this beta cell from iPS, to work with experts in immunology like Bart to see how we can also dampen the immune pressure in order to get this cell to survive. Another way is also that something that we are working on is to genetically modify the beta cell prior transplantation to make them less prone to be destroyed by T cells. So, but all these cellular therapy are extremely promising, but they are still, not on the market on the short term, let's say.
I have another question, and that relates to research being done by JDRF elsewhere in the Netherlands, and that has to do with a shark's cage. Remember that things cannot come in, but things can come out, or so the safe way. Is that also what you're thinking of when you think of modified beta cells that should be implanted into a body?
Yeah, so absolutely. So there is some. So I think you're referring to the work that is performed, I think, in Maastricht, I think, where indeed the idea is to, since all these cells that will be re-implanted in the body from a diabetic person, to protect them with a kind of, if we can make a picture of the device. It's kind of a tea bag in which you will find this islet, and this bag will protect the beta cells that are inside from the infiltration of the immune cells. So this is kind of, without talking about genetic manipulation of the beta cells, is a kind of holy grail that people are working on at the moment to try to have a device that will allow glucose to enter and insulin to be secreted without getting access to the immune cells. And this is, I think, very difficult challenge, but I think the works are very promising. So to summarize, I mean, two aspects to protect the beta cells, or this device, or the genetic manipulation.
And if you put it on a timeline, of course, in 10 years of JDRF in the Netherlands, you actually are very surprised when you look back and you think, okay, so what have we done? We've done artificial pancreas. We've managed to get continuous glucose monitoring not only available, but also even reimbursed by insurance. We have the nasal glucagon. So there is a lot of progress happening. And even though people are listening here like, but how close is a solution? If you put it on a timeline and have to say, well, one of them I think we'll have within 5 or 7 or 10 years, where are we? And what do you think you'll see first? And that includes postponing triggers to come. So that's actually Bart Roep's work, I would say, and his vaccine story, which we had last year. But yours as well. If you catch it soon enough, what could you do? And how long do you think? Because you're already testing on patients. How long do you think before it becomes available to that little group, but to a group?
Yeah, so I think to come back to this timeline, I think the first development in type 1 diabetes treatment are, of course, the monitoring and indeed the better blood glucose control. What we are doing by identification of this small peptide is also very useful. I mean, we are trying to give some tools to Bart, who is developing incredible technology to make this vaccine. So to make a long story short, what he's trying to do is trying to re-educate or retrain the immune system to be tolerant to some protein in a kind of vaccine. And by identification of this error, what we do, Bart can also continue our work, at least we work always together in this kind of aspect, is to try to retrain the immune system to accept the errors that we have identified. So we are trying to, as much as we can, to work hand in hand to try to see whether we can combine or join forces in this aspect. So I think what will come on the first, it will be so again, the work of Bart is already, and the phase 1 has been done last year. The phase 2 will start. So I think this kind of vaccine or retraining of the immune system will come very soon, I think. Regarding drugs to relapse the stress, I think it can go very fast, but in the same time, we need to be aware of the risk benefit when we talk about a drug treatment. And I think it's a very difficult aspect when we are talking about type 1 diabetes, because insulin is available. And we need to be aware that if we have a drug that will have to be taken also on a daily basis, but with more side effect. So I think it's this kind of balance when we talk about a drug, that the safety is very, very, very important, even more for type 1 diabetes than for other diseases, because there is insulin. So
do you think people have learned quite how medicine and approval of treatment works, thanks to having seen how coronavirus vaccines were being managed and how careful the authorities are?
I agree. And there is one, if I can add one word about the COVID, I think it's when we were in our project, when we talk about understanding for a cure, I think the discussion about COVID is the very good example. It's exactly the example of if you know how a disease occur, you can cure that. Yes. We know from history, we know almost everything about coronavirus derived, family. We know exactly how the virus enters a cell. We know exactly what is expressed at the cell surface. And you see that with this COVID story, so of course it was a terrible period, but the you can also illustrate that if you know exactly how things develop, it took basically a couple of months to get a vaccine against corona because you know how it works. In type 1 diabetes, if we don't know how it works, it's extremely difficult to find the cure.
Yes, because remember, let me remind the audience that actually all the scientists we have on the program keep reminding us there is not just one type 1, there are many types of type 1. But I think that what you were analyzing here is that if you determine the root cause analysis, it changes the way that you look for medicines, for a cure. And I think that is the major finding and the major realization moment in your research is, guys, we've been working on the wrong thing. We've been working on all kinds of different ways to treat this, but actually nobody has been paying attention to finding a way to manipulate the beta cells. Is that correct?
Yeah, true. And to continue on this COVID episode, I mean, you were talking about the authority that was also very open to try new strategy. I think so. It was also very beneficial. But we also need to realize that with all the development that has been made, not, for example, this mRNA vaccine that was very quickly on the market, whereas it has been stuck for years with the authority, and it has been proven that it's been very efficient. I mean, we can also benefit from all this research that has been done on this mRNA vaccine. Why not in the future have an mRNA vaccine against type 1 diabetes in a similar fashion? So I think this we have learned a lot with the COVID and how indeed our fast research can go when we understand the process. And we have learned also a lot on new development from the COVID. And I think it will also be beneficial for type 1 diabetes and for so many other diseases. We're already talking about mRNA vaccine for tumor, for example. So it has opened a new era, I think, for treatment of many diseases.
Yeah, so it's a blessing in disguise, maybe. But if I look at the questions here, and I look at the questions that were submitted before the webinar, one of the questions that people are asking right now is, what can they do in the meantime? You already said, make sure your blood glucose is as stable and as good as possible. Everybody knows where it should be, but you have an extra reason now because you've shown pictures of what actually happens when you're creating stress inside your beta cells. But the second thing is, what else can people do? Vitamin D3. Is that something, I mean, this is coming from your combined research, right? Mm-hmm. So this is one of the things that Bart was saying. Remember, take it. I wouldn't say baat het niet, gaat het niet, but everybody knows that little kids don't get enough sunlight, don't get enough whatever, and you should be using this. What else can people do? And what should they be waiting for next?
So I would say that I study in Nice, so go to Nice to get some sun, maybe. So, but as a job, but indeed vitamin D is indeed an important factor. I would say we discussed, I think, a lot about the fact that type 1 diabetes was an autoimmune disease, is an autoimmune disease, involves stress, but also we should not forget that it's also a metabolic disorder, and the glycemia also have a large impact on the beta cell viability and also on beta cell stress. So I will also mention again that a proper blood glucose control and for example, also will reduce the stress. So there are lots of studies going on at the moment on the change in diet in order to maybe reduce the glucose intake. Because of course, when you have a high glucose, it means that the consequence, there is a high insulin secretion and potentially a source of stress. You can imagine that if you have a regimen or diet with less carbs, the pressure on this beta cell can be less and will help maybe to prolong, I think, the beta cell life. So there are lots of studies.
This is exactly what's going on in the chat right now. Yeah. So yes, questions about slim A to ND8. You can read back to people who aren't in the chat of this call. There's a lot of conversation on that now.
But I will add one more thing, if I may. So the diet are possibly or extremely interesting, I think, to reduce the glucose intake. But again, I mean, as a diabetic, you should, if you have the idea to change drastically your diet, you always have to be in relation with a clinician that will monitor the disease and monitor the blood glucose, monitor other parameters to be sure that you're not entering in some worse disease, because I mean, we have also report of ketogenic diet inducing dyslipidemia, inducing also ketoacidosis. So it's lots of things that you need to take into account when you change the diet. So that's why the implication of a physician in a close discussion is very important when you talk about diet.
Yeah. So that's obviously a very good disclaimer. Don't sit there and think, okay, I got all my carbs. Talk to your doctor. Tell them that you're planning to do this. That's a good disclaimer. A lot of people are now asking, and they have been asking, what can I do myself? Let me summarize what you said. You said, avoid, keep eating healthy. Make sure your blood sugar is as good as possible. Parents are saying, but what about my kids? They need to grow up. They need their carbs. They need fuel. And of course, all of us know there's a difference between good and bad carbs, but could you say something? Because people are confused now. What can they do?
So again, another disclaimer, I'm not a dietitian, so I think it's also a bit difficult for me to really give advice on that aspect. But I recognize that it's extremely difficult for parents with a teenager. I can imagine it's a very difficult situation to control all the food intake. Again, that's what I just mentioned. A discussion with a clinician, discussion with a diabetologist, discussion with a dietitian is very important for this parameter.
Yeah, I'm watching people say, all right, I'm hoping, I keep on hoping that I'm going to hear that we're going to get a cure very soon, right? The fact that you say, listen, the fact that I've discovered that we were looking in the wrong place, or we should actually look here, means that there is progress. You can't jump on it all together, but you are showing that with a new root cause analysis, a different kind of treatment should be able to be available. So do you have a timeline for us?
Yeah. So the timeline is always a difficult question, and it's we need to give hope, but we have to be also fair in what we say. But I mean, it's not because we have found this the fact that the beta cell participate to the disease that all the therapy that are focusing on the immune system are completely useless. What I mean is discussion we had with Bart many times. Probably the combinatorial treatment is something that you can envisage in the future. So again, targeting the immune system with strategy developed by Bart, combining with maybe a drug that will relapse stress, or maybe combine with a special diet that will relapse the stress. So again, it's not that the treatment are not there. It's we need to find a way probably to combine these two aspects to have an ideal treatment. This being said, it's also very important to realize that you may have conclude yourself at the presentation. There is different cause for type 1 diabetes. There is different type of diabetes. There is people that can manage their glucose very well, some that cannot do that. So there is a very large heterogeneity in the type 1 diabetic patient. So we need also to be aware that probably the magic bullet that will cure all the type 1 diabetes is probably something that we may, at least we may forget and see more, that we need to have a proper diagnostic of the patient to offer something of personalized medicine. In that respect, it will mean that several treatments should be envisaged for the type 1 diabetes.
Absolutely. And you already mentioned that as we see this, there is also more convergence with type 2. So several things might, it might just make a bigger population for you to work on, and therefore easier to develop a medicine.
Yeah, you're absolutely right. I mean, going back to what we just said, that if type 1 diabetes, again, being a metabolic disorder, in taking into account all the research that has been done on type 2, all the investments from the pharma company developing drug to help people with type 2 diabetes to manage their glucose, all these drugs can then be redirected to type 1 to evaluate their efficacy again on certain group of patients, whether they have an effect in maintaining beta cell function by having a proper blood glucose regulation. So we have, by bringing type 1 and type 2 close together, we also have the opportunity to use all the drugs that have been used for type 2 diabetic patients.
Yeah, what you're actually saying is the awareness is there now, and there is kind of a rush, because now suddenly people are saying, what you mentioned thanks to COVID is that the same type of sequencing is relevant now, and there is kind of like a speed lane that we're finding. That's the impression that I get. Yes. And I think that's also what the audience needs to hear, that together now you and Bart, in your own two sides of the trigger for type 1, have suddenly gone into warp speed. You're discovering different things, and suddenly things become a lot easier, a lot more easy.
Yeah.
How would you summarize for today?
So I summarize that maybe it's I can conclude with the same word I started with. I mean, the network of people, of scientists from different fields, from different expertise, like we've done with Bart being an immunologist, me as a poor immunologist but molecular biologist, by sharing expertise. But we need also to put into this group of scientists, also endocrinologists, clinicians, and JDRF have the capacity to bring everyone around the table. And with this, it's with this kind of strategy that we can also exchange and, for example, learn from different diseases, learn from different expertise. And this, I think, hasn't been done before, probably because it was thought to be only one aspect. And let's try to focus on this single aspect. If now we realize that the type 1 diabetes is a problem, an endocrinology problem, a metabolic disorder, an autoimmune disease, a virology problem, because we know that viruses are also implicated as a trigger of type 1 diabetes. Suddenly, you will have expertise from different people, and I think it's this joint network, I think, initiated by JDRF that makes the speed of research faster in the near future.
Yeah. There are so many questions coming in. I see is Big Pharma even interested in real solutions? I am seeing, what about the psychological stress of, for instance, exams? And you keep on saying it's a circle. If your blood glucose is not okay, then your other hormones will start playing up. Then you will have more stress. So it's not the beta cell stress that you feel during your exams, but it's a cycle that makes it. And I think that's what we need to keep saying. I'm seeing the conversation, the chat is going like this all the time. And meanwhile, I have over 60 questions that are all here. I think what we'll just have to do is collect the questions, make sure that you've already seen many of them. They're about the triggers. They're about the stress. I think people now understand that the stress is not the stress due to whether you pass your exam or not. But of course, when you have type 1, the cycle that you go in when you're worried will help. But it's not that kind of stress that you meant when you said inflammation and therefore cell stress.
Yeah, a bit also. I mean, again, we are not equal facing stress. It's unfortunately, but all kind of stress have for consequence an increase of your blood glucose. So it's not the direct effect necessarily of the stress on the beta cell, but it's the consequence of the stress on the metabolism of the beta cell, for example. And if you have an increased blood glucose, you have a source of stress. Similarly, again, viral infection. It has been shown there are lots of correlation with viruses without any sign of direct infection with the virus, but the consequence of an infection triggers an increased blood glucose. And there are lots of consequences of this kind of outside event that participate to a change in the glucose homeostasis that is a source of stress.
There was one final question that just came in. Is there a way to destroy the wrong sequence?
I'm not sure that there is a way to destroy the wrong sequence. I mean, the immune system does it. Maybe what we were aimed to is to prevent the formation of this mistake. So instead of destroying it,
you're smart. That is indeed the root cause analysis that we keep asking you to do. Thank you so much. Yes, Ingrid, I think we have so many more questions that we could easily fill another hour. But I think that if we have in people's mind, this is a different way of looking at root cause analysis. The cell biologists are looking at, and you've been doing this for 5 years, so you're really sure. After the trigger, what you should be doing. I think that we all now understand that you would be working on a different kind of drugs. And you have again reminded people how important it is to keep your blood glucose where it should be, because it really does matter. I think we've shown what the essence is of the progress you've made. And we've also shown the essence that you touched of the systematic and various strategy approach that JDRF is doing by having both you and Maastricht and so many other universities worldwide actually working together to find what is going on. And of course, your close cooperation with Bart Roep is something that everybody at JDRF understands and very much appreciates. So Ingrid, I hand over back to you because I think there are too many questions now and we're all in the same direction. I think we have it now.
Yeah, thank you so much, Els. Thank you so much, Arnaud. Bring everyone around the table. That's the message. There is so many knowledge, and how can we combine that knowledge towards the solution? And I think also it was a very nice sentence what you used, Arnaud. Finding a cause is finding a cure. So the cause is necessary to find a cure. So thank you so much, all of you, all of the visitors. I have a few questions after the webinar. Please, for all the people, all the viewers, please fill in 5 questions so we can do the next webinar even better. Thank you for everything for now. I hope everybody will join the 19th of June. Bart Roep will be there also when it's possible in the hour of this, the scientific researchers. So thank you all. And I wish you a very good evening. And we will make a blog. We will make some information about all what is said tonight, and also all the questions what was open for now. So thank you.
Thank you very much.
Thank you very much. Have a wonderful evening.
Have a wonderful evening. Thank you.
Thank you.